Did you know that if you could transport a farmer from the year 1000 CE into your kitchen today, they wouldn't recognize a single thing in your refrigerator? Not the corn, not the bananas, not even the carrots. What we call natural food is actually the result of a 10,000-year genetic engineering project conducted in fields across every continent. Today, I'll reveal the 15 most dramatically transformed foods in your kitchen. Everyday fruits and vegetables our ancestors engineered over hundreds of generations using nothing but patience and observation. Number 15 would be toxic in its original form. It's in your kitchen right now. Stay to the end, because the final transformation will change how you think about natural forever. Let's begin. Number 15. Corn. Imagine walking through Mexican highlands 9,000 years ago. You spot a wild grass barely knee-high, its seeds encased in hard shells like tiny nuts. This is teosinte, corn's unrecognizable ancestor, producing maybe 5 kernels smaller than rice grains, bitter and tough. Now picture the golden ear you shucked last summer. Hundreds of plump, sweet kernels in perfect rows. The transformation from teosinte to modern corn represents one of agriculture's most dramatic genetic shifts. Scientists at the University of Wisconsin identified that just 5 major genes control the difference. But those genes required thousands of years of selection to unlock. What the ancient Olmec and Maya accomplished seems almost impossible. They took a grass offering nothing and saw potential written in its DNA. Generation after generation, they saved seeds from plants producing slightly larger kernels, softer shells, more seeds per stalk. They couldn't sequence genomes, but they understood heredity with an intuition bordering on mystical. Here's what most don't realize. Modern sweet corn contains a mutation discovered in the 1700s that prevents sugars from converting to starch. Every ear carries this deliberate genetic alteration, kernels arrested in an immature, sugar-rich state, forever sweet. The most disturbing part? Modern corn literally cannot reproduce without us. Drop an ear on the ground, and it rots. The kernels are packed so tightly they'd strangle each other if they sprouted. We created a crop that would go extinct within a generation without human intervention. Corn isn't just food. It's a 9,000-year partnership between human ambition and plant potential, written in genes we couldn't see, but somehow learned to rewrite. Number 14. Bananas. Picture the original wild banana. Seeds the size of gravel fill its thin flesh, making each bite a dental hazard requiring strategic spitting. The fruit barely reaches finger length, tasting vaguely of chalk and bitterness. This is Musa acuminata, the Southeast Asian ancestor of every banana you've peeled. The transformation happened through a genetic accident humans learned to perpetuate. Around 10,000 years ago in New Guinea, certain banana plants experienced polyploidy. Chromosomes tripled instead of doubling. The result was a sterile mutant producing seedless fruit, unable to reproduce sexually, dependent entirely on humans propagating through cuttings. Modern Cavendish bananas are all clones, every single one genetically identical, traced back to a specimen discovered in 1836 in the Duke of Devonshire's greenhouse. You're not eating a species, you're eating a botanical photocopy repeated millions of times across 4 continents. Here's the crisis. Panama disease TR4, a fungal plague, is spreading across plantations worldwide. Because every Cavendish shares identical DNA, They share identical vulnerabilities. Scientists warn we may lose the Cavendish by 2040. Exactly what happened to its predecessor, the Gros Michel, destroyed by a different strain in the 1950s. That banana you grabbed this morning, convenient, seedless, perfectly curved, represents humanity's greatest agricultural triumph and potentially its most fragile failure. We created a fruit so perfect, it forgot how to survive. Number 13. Watermelon. Travel to the Kalahari Desert 5,000 years ago. You crack open a round fruit the size of a tennis ball, hoping for relief, and find pale white flesh threaded with bitterness and seeds outnumbering edible parts. This is the tsama melon, watermelon's barely recognizable ancestor. Ancient Egyptian tomb paintings from 2000 BCE show the transformation underway. Melons with reddish flesh, though still dominated by seed cavities. The Egyptians selected generation after generation for redder flesh, more sugar, less bitterness, unknowingly breeding for lycopene production, the same compound making tomatoes red. By the 1600s, European paintings show watermelons closer to modern versions, but with a distinct radial star pattern and pale center. Uniform sweetness throughout came in the early 1900s through systematic hybridization. Here's the laboratory miracle. Seedless watermelons exist because of chromosome manipulation pioneered in 1939. Japanese scientists used colchicine to double watermelon chromosomes, creating tetraploid plants. Crossed with normal diploids, the offspring are sterile triploids, genetic dead ends producing fruit, but never viable seeds. Those tiny white seeds in seedless watermelons aren't seeds. They're empty seed coats, botanical ghosts of the reproductive system we deleted. That watermelon in your refrigerator, uniformly red, impossibly sweet, conveniently seedless, required 5,000 years of selection and 20th century genetic manipulation. Nature made a bitter survival tool. Humans made dessert. Number 12. Carrots. Imagine Afghanistan in the 10th century. You pull a carrot from soil and discover not cheerful orange, but a purple or white finger-sized taproot. Thin, woody, intensely bitter from terpenes, evolved to deter insects. This is the original carrot that would make you wince. The transformation to orange happened through Dutch patriotism. In the 17th century, during the House of Orange's rise, Dutch farmers systematically selected carrots with higher beta-carotene, the pigment-producing orange color. What started as honoring their royal house accidentally created one of agriculture's most nutritious vegetables. Modern carrots contain 25 times more beta-carotene than wild ancestors. Here's the biochemical revolution. When we engineered carrots for sweetness, we altered their defense chemistry. Wild carrots produce bitter terpenes protecting against fungi. Domestic carrots still produce these, but in dramatically reduced quantities, trading chemical warfare for palatability. For most of human history, carrots weren't eaten for roots at all. Greek and Roman physicians prescribed carrot seeds as medicine. The root was animal fodder, too bitter for refined palates. That baby carrot you dipped in hummus? It's been genetically engineered over centuries to bear almost no resemblance to the bitter, purple root wild plants still produce. Carrots aren't naturally orange. They're naturally armed. We convinced them to lower their weapons. Number 11. Cabbage, broccoli, kale, cauliflower, Brussels sprouts, and kohlrabi. Here's something that will alter your understanding of vegetables. Cabbage, broccoli, kale, cauliflower, Brussels sprouts, and kohlrabi are not different species. They're all the same plant, Brassica oleracea, sculpted into different forms through selective breeding. The wild ancestor still grows along Mediterranean coasts, a gangly, bitter plant with small leaves and unremarkable flowers. Around 2,500 years ago, different cultures began selecting for different traits. Ancient Greeks selected for larger leaves in tight heads, creating cabbage. Romans found plants with thick stems and selected for that, creating kohlrabi. Medieval Europeans discovered tiny heads growing up the main stem and cultivated Brussels sprouts. Here's where it gets strange. Broccoli and cauliflower are arrested flowers. The tight heads you eat are thousands of flower buds locked in an immature state, bred to never open. We're eating botanical fetuses, flowers that will never mature. When broccoli does flower, if left too long, it suddenly reveals its true nature. The tight head loosens, and small yellow flowers emerge, identical to wild mustard. Genomic studies confirm the impossible. Despite looking completely different, these vegetables share over 98% of their DNA. Visual differences come from changes in a handful of regulatory genes. We didn't just breed vegetables, we sculpted a single plant into 6 different forms, like botanical origami written in DNA. Number 10. Strawberries. Imagine walking through European woodlands in the 1200s. You spot tiny wild strawberries, fraises des bois, barely fingernail-sized, intensely fragrant, but producing so little flesh, you'd need hundreds to make a single modern mouthful. These diploid berries have just 2 sets of chromosomes. They're what strawberries were meant to be. Delicate, ephemeral, more perfume than fruit. The transformation required a genetic accident on a continental scale. In the 1700s, French spy Amédée François Fraisier smuggled Chilean strawberry plants to Europe. When planted near Virginian strawberries in a Brittany garden, something unprecedented happened. The Chilean and Virginian strawberries cross-pollinated, creating genetic freaks. Octoploids with 8 sets of chromosomes instead of 2. Like a human suddenly having 4 biological parents contributing DNA. The resulting hybrid strawberries were enormous, with flesh firm enough to ship and sweetness eclipsing their ancestors. Here's the genetic absurdity. Every commercial strawberry descends from this 18th century botanical accident. Modern strawberries contain 4 times the genetic material of wild ancestors, polyploid monsters with over 35,000 individual genes, more than humans possess. Those octoploid strawberries are so far removed from wild ancestors, they can barely interbreed with them. We've created a genetic island, a fruit existing in reproductive isolation of our own making. Strawberries aren't evolved. They're assembled, stitched together from chromosomes that should never have met. Number 9. Tomatoes. Picture Peru 500 years ago. You encounter a wild tomato plant with currant-sized fruits, intensely yellow-green, filled with tomatine and solanine that make them mildly toxic. The Inca didn't eat them, they used them as ornamental curiosities, pretty but dangerous members of the nightshade family. When Spanish conquistadors brought tomato seeds to Europe in the 1500s, the plant was classified as poisonous. They weren't entirely wrong. Wild tomatoes produce high levels of alpha-tomatine that can cause nausea, vomiting, neurological disturbances. Italian farmers saw past the poison. They selected plants producing less tomatine and more sugars, slowly breeding toxicity down to harmless levels. By the 1700s, tomatoes had become pomodori, golden apples, and Neapolitan cuisine made them indispensable. Here's what we sacrificed. Modern commercial tomatoes contain the RIN mutation, ripening inhibitor, preventing the fruit from softening normally. These tomatoes ship across continents, get gassed with ethylene for cosmetic redness, and taste like barely flavored water. We traded flavor for logistics. Recent studies revealed that when we selected for larger fruit size, we accidentally reduced nutritional density. Wild tomatoes pack nearly 10 times the vitamin C and flavonoids of commercial varieties. We didn't just domesticate tomatoes, we lobotomized them, selecting for beauty and shelf life while accidentally breeding out complexity and nutrition. Number 8. Apples. Imagine Kazakhstan's forests 50 million years ago, where wild apples, Malus sieversii, evolved. These fruits were small, intensely sour, sometimes bitter, filled with tannins designed to deter browsing animals while enticing birds to spread seeds. Apples traveled the Silk Road with traders who selected rare sweet fruits, propagating them through grafting. By Roman times, over 30 named cultivars existed. When English colonists arrived in America, they discovered something strange. Planted apple seeds rarely produced fruit like their parents. This reveals apples' dirty secret. They're extreme heterozygotes. Plant an apple seed, and it will likely produce inedible fruit. Every Honeycrisp, Granny Smith, and Fuji exists because someone grafted a branch from one specific tree onto rootstock, cloning it endlessly. That Honeycrisp isn't a breed. It's a cutting from a single Minnesota tree discovered in 1960, now duplicated millions of times. Modern dessert apples have been selected for mutations increasing fructose while decreasing malic acid. Wild apples contain up to 10 times more acid, sour enough to corrode tooth enamel. Commercial agriculture has narrowed apple diversity to about 10 major cultivars. Meanwhile, the USDA preserves over 2,500 varieties carrying genes for disease resistance we may desperately need. Apples aren't grown anymore. They're photocopied. Each tree a genetic replica that would die within seasons if left to defend itself in the wild. Number 7. Wheat. Picture the Fertile Crescent 10,000 years ago. You encounter wild grain called einkorn, tall, brittle stalks that shatter at the slightest touch, scattering seeds before you can harvest them. Seeds encased in tough husks require laborious processing. Each plant produces maybe 20 to 30 seeds. This is wheat's miserable ancestor. The transformation happened through agriculture's most important accident. Ancient farmers noticed rare mutant plants where seed heads didn't shatter when mature. This mutation, a change in just 2 genes, meant seeds stayed on the plant until humans were ready. It seems small. It's actually the genetic foundation of civilization. Here's where wheat's story becomes science fiction. Modern bread wheat, Triticum aestivum, is a hexaploid containing 6 complete chromosome sets from 3 different grass species. This genomic complexity contains over 100,000 genes, 5 times more than humans possess. Norman Borlaug's Green Revolution varieties in the 1960s selected for semi-dwarf wheat with shorter, stronger stems that wouldn't fall over under enlarged seed heads. The result? Wheat dedicating more energy to seeds instead of stalks, doubling yields. Modern commercial wheat has been selected for uniformity to the point of vulnerability. Scientists race to reintroduce resistance genes from wild wheat relatives, diversity we deliberately bred out. We didn't just domesticate wheat. We assembled it from incompatible genomes, then redesigned its architecture. Number 6. Peaches. Travel to Western China 8,000 years ago. You find a wild peach barely larger than a cherry, with thin, sour flesh clinging to a massive pit comprising most of the fruit. The flesh-to-pit ratio is dismal, maybe 20% edible to 80% stone. This is Prunus mira, more seed dispersal vehicle than food. The transformation happened remarkably fast. Archaeological evidence shows that in just 3,000 years, peaches doubled in size and increased their flesh-to-pit ratio to 70%. Genomic studies identified that genes controlling cell division in fruit flesh underwent intense selection, essentially telling the peach to keep growing flesh while keeping the pit constant. Wild peaches produce high levels of amygdalin in their seeds, the same cyanogenic compound in bitter almonds. Crack open a wild peach pit, and the kernels taste intensely bitter, with enough amygdalin to make you sick. The texture transformation is equally dramatic. Wild peaches have flesh barely separating from the pit. Clingstone. The development of freestone peaches, where flesh easily separates, involved mutations in genes controlling pectin degradation. That peach you bit into, juice running down your chin, pit sliding free, represents 8,000 years selecting for a single trait. More food, less seed, maximum pleasure per bite. Number 5. Lettuce. Imagine the Mediterranean 5,000 years ago. You encounter wild lettuce, Lactuca cereola, standing nearly 4 feet tall, covered in prickles, with thin, bitter leaves producing milky latex when broken. This latex contains lactucarium, a compound with mild sedative properties that ancient Egyptians called lettuce opium. The ancient Egyptians cultivated it for the stem and latex, not leaves. Tomb paintings from 2500 BCE show lettuce with elongated stalks. Around 2,000 years ago, Romans started selecting for larger, less bitter leaves and reduced latex production, unknowingly selecting against sesquiterpene lactones that made wild lettuce medicinal but unpalatable. The diversity is stunning. Butterhead, romaine, iceberg, leaf lettuce, all the same species sculpted into different forms. Iceberg lettuce was developed in the 1940s specifically to survive cross-country shipping. Lettuce redesigned as packaging material for itself. Different lettuce varieties show wildly different nutrient profiles. Dark red and purple lettuces contain up to 10 times more antioxidants than pale iceberg. When we selected for mild flavor and pale color, we accidentally selected against nutrition. That salad you ordered, crisp, mild, refreshing, descends from a tall, prickly weed producing narcotic sap. We didn't domesticate lettuce. We drained it, breeding out almost everything that made it interesting or nutritious. Number 4. Almonds. Picture Central Asia 5,000 years ago. You crack open a wild almond and taste shocking bitterness followed by numbness. Within minutes, you feel dizzy, nauseous. You've consumed amygdalin, which your body converts to hydrogen cyanide. Wild almonds are poisonous. Eating 10 to 15 could kill a child. Here's the genetic lottery that changed everything. Somewhere in ancient orchards, a mutation occurred in a single gene, SK1, that triggers amygdalin production. This mutation created sweet almonds, disarmed almonds that forgot how to make poison. Ancient farmers carefully propagated this through grafting, cloning the mutation across generations. The biochemistry is elegant. Wild almonds produce amygdalin, as defense against seed predators. When you bite one, enzymes break it down into glucose, benzaldehyde, almond flavor, and hydrogen cyanide. Sweet almonds still produce trace amounts. You taste the benzaldehyde, but cyanide production is reduced by over 99%. Sweet almonds are so rare in nature that every commercial almond tree is grafted. Plant a sweet almond seed, and there's a 50% chance the resulting tree will produce bitter, poisonous almonds. The mutation is recessive, and wild genetics constantly threaten to reassert themselves. That handful of almonds you snacked on, mild, slightly sweet, perfectly safe, represents one of agriculture's most dramatic detoxification projects, a 5,000-year effort to convince a poisonous seed to disarm itself. Number 3. Eggplant. Picture India 4,000 years ago. You encounter a plant bearing small, round fruits covered in spines, ranging from white to purple. The fruits taste intensely bitter, almost acrid, filled with solanine causing nausea if eaten raw. This is wild eggplant, Solanum incanum, a thorny, defensive nightshade family member that dares you to eat it. Indian farmers selected rare plants with fewer spines, larger fruits, and reduced bitterness, unconsciously breeding for mutations in genes controlling alkaloid biosynthesis. By 300 BCE, Sanskrit texts describe cultivated eggplants in Indian cuisine. Here's the bizarre diversity Western eaters miss. Eggplants come in dozens of shapes, sizes, and colors. Tiny Thai pea eggplants, long Japanese varieties, white egg-shaped fruits, giving the plant its English name. The deep purple globe eggplant represents just one cultivar among hundreds. That glossy purple skin contains nasunin, an anthocyanin with potent antioxidant properties. When breeders selected for deeper purple, they accidentally concentrated beneficial compounds. Meanwhile, the flesh was selected for reduced bitterness by decreasing solanine, disarming the plant's chemical defenses. Arabs introduced eggplants to Europe in the 13th century, but Europeans remained suspicious for centuries, calling them mad apples and believing they caused insanity. We didn't just domesticate eggplant, we negotiated with it, developing cooking methods to overcome bitterness while breeding varieties needing less convincing. Number 2. Grapes. Travel to the Caucasus Mountains 8,000 years ago. You find wild grapes, Vitis vinifera sylvestris, with tiny berries barely pea-sized, intensely sour, filled with large seeds and thick, astringent skins loaded with tannins. These grapes exist to be eaten by birds, not to please humans. The domestication happened simultaneously with wine invention, probably not coincidentally. Early farmers discovered that fermenting these sour fruits produced revolutionary alcohol that didn't spoil, could be stored, and produced euphoria. Selection immediately favored grapes with higher sugar content. More sugar meant more alcohol, and larger berries easier to process. Modern genomic studies show domesticated grapes underwent selection in genes controlling flower sex. Wild grapes are dioecious, domestic are hermaphroditic. Seed size, skin thickness, sugar metabolism, and berry size. We redesigned the entire fruit. The diversity is staggering. Over 10,000 named cultivars exist worldwide. Seedless grapes exist through a mutation called stenospermocarpi, where seeds start developing but abort early. Nearly all wine grapes are grafted onto American rootstock because European varieties proved devastatingly vulnerable to phylloxera, a root aphid that nearly destroyed European wine industries in the 1860s. That grape you popped in your mouth? seedless, sweet, crisp, required genetic manipulation to create sterile seeds, centuries of selection for sugar content 10 times higher than wild varieties. Number 1. Potatoes. Picture the high Andes of Peru 10,000 years ago. You dig up a wild potato, Solanum brevicole, and find a small, bitter tuber riddled with glycoalkaloids, particularly solanine and chaconine, producing a burning sensation in your mouth and gut. Eat too many raw wild potatoes, and you'll experience nausea, headaches, neurological symptoms, even paralysis. This is what potatoes were meant to be: protected, toxic, nearly inedible. The Inca accomplished something extraordinary. They developed over 4,000 distinct potato varieties and learned to process toxic ones through freeze-drying, leaving potatoes out to freeze at night, then trampling them to squeeze out moisture and glycoalkaloids, creating chuño that could be stored for years. They were detoxifying potatoes through engineering. When Spanish conquistadors brought potatoes to Europe in the 1500s, they brought only a handful of lowland varieties. These underwent intense selection for reduced toxicity, larger tubers, and better flavor. Modern potatoes contain about 90% less glycoalkaloids than wild varieties. We literally bred the poison out over 400 years. Here's the continued danger. Potato plants still produce glycoalkaloids in response to light exposure and stress. That's why potatoes turn green when exposed to light. The green chlorophyll accompanies solanine production. A potato that's green enough to taste bitter contains enough solanine to make you sick. What the Irish famine of 1845 taught us harshly. The European potato population descended from a tiny genetic bottleneck, making them devastatingly vulnerable to potato blight. Over 1 million people died because our domesticated potatoes lacked disease resistance diversity that Andean varieties possessed. Recent genomic studies revealed that modern potatoes have lost approximately 75% of the genetic diversity present in wild Andean populations. Every trait we selected for came at the cost of stress tolerance and disease resistance. That baked potato you ate last night, fluffy, mild, comforting, descends from a toxic tuber that would have made you seriously ill. 400 years of selection have convinced it to mostly disarm, but the genes for poison production remain, dormant but waiting, ready to activate if we stop paying attention. Potatoes aren't naturally safe, they're naturally armed. We've negotiated a fragile truce, but the capacity for violence remains written in every cell, one stress response away from reasserting itself. So there you have it. 15 foods proving natural is a word we should use carefully. From corn that cannot reproduce without human hands, to bananas that are all clones of a Victorian specimen, to almonds we convinced to stop producing cyanide, every item reveals a truth. There is almost nothing natural about the food in your kitchen. We didn't just domesticate these plants, we rewrote them. Over 10,000 years of selection, grafting, and lucky mutations, humanity has sculpted wild plants into forms unrecognizable to our ancestors. We've created seedless fruits, toxin-free vegetables, grains that can't disperse their own seeds. The most humbling part? This happened without laboratories, without understanding DNA. Ancient farmers worked with nothing but observation and patience, and achieved genetic transformations as dramatic as anything modern science has accomplished. But here's the question: what have we lost? Every selection for sweetness, size, or convenience came with trade-offs. We bred out bitterness and accidentally removed medicine. We selected for beauty and lost nutrition. We chose uniformity and created vulnerability. The foods on your table tonight are living museums of human ingenuity. They're also warnings about genetic narrowness and the fragility of our food system. Next time someone tells you to eat natural, smile, and remember. There's almost nothing natural about food anymore. We've been genetic engineers for 10,000 years. We just used fields instead of laboratories. If this video changed how you see your grocery store, smash that like button and hit subscribe. Next week, 15 foods that would kill you in their original form. Drop a comment telling me which transformation shocked you most. See you in the next one.